Okay, so welcome to this first video lecture in course 2851, Principles of Metabolism at Karolinska Institutet. So this is an introduction lecture, so we'll be covering a bit of overview stuff, general things, and sort of prepare for the stuff we'll do in the remaining lectures in this course. So let's get started. So let's start very simple. What is cellular metabolism? So we'll be looking at metabolism mostly on the cell level in this course, not so much the whole body level with all the complications with multiple organs and things like that. We'll be studying cells. And at the level of cells, metabolism is really the transformation of nutrients. So in Greek, metabolism means change, and metabolism is changing molecules into other molecules. So this is very basic, obviously. You've all seen this, but it's useful to think about what kind of metabolic tasks that a cell has, and it differs a bit between cell types. So all cells, of course, need to take up some form of nutrients. This is usually sugars, fats, amino acids, and they process them in various ways depending on what kind of needs they have. So some cells have large energy needs. They might be cells that do a lot of work, like muscles that contract or neurons that have to fire action potentials or things like that. So they might have a large energy need, and they will use a lot of the nutrients to process them into energy to be able to do that work. But all cells have some kind of basic energy needs for their sort of self-maintenance. Cells need to maintain ion gradients, synthesize proteins to keep themselves intact and things like that. So there's always some energy metabolism going on. But then it kind of diverges between cell types. So some cell types are very biosynthetic, like producing antibodies like this one. A B cell would do that. Fibroblasts, for example, produce a lot of protein. Adipocytes, of course, produce a lot of fat. So these cell types might be specialized and produce a lot of a particular thing, and then their metabolism will be geared towards that. And some cells are proliferating, so they produce new cells, and that's of course very complex. You have to reproduce the entire biomass and pretty much all the structures in the cell. And for that, the metabolism becomes quite complex and very interesting, really. This is what we study a lot in my lab. And then you have to produce proteins and fats and lipids and nucleotides and all sorts of things. So it can vary a lot depending on cell type. And then there's always some waste. So no cell has a perfect metabolism where everything in the nutrients can be taken care of and used. There's always something that goes to waste. So that's pretty much what it looks like, and this is what we're going to be studying in this course. And of course we'll be looking at the inside here, the box that does all of this. And of course this is very complex, and this is largely why metabolism can be difficult. So you've all seen these wall charts, these large sort of posters with thousands of things on them, and there are enzymatic reactions going in all directions, and it can be very sort of overwhelming. We tend to think about this nowadays as metabolic networks in the sort of era of systems biology. So we think about this as a large kind of network, a graph with enzymes and reactions and things connected together. And there are systematic tools for analyzing this, but still, grasping this, conceptual level can be very difficult. So we'll try to get to some tools for doing this, for being able to grasp this system. So one sort of conceptual tool, a very common concept, is the pathway. And this can be very helpful if you study a particular system in isolation. If you want to study the synthesis of one thing, say a purine, a nucleotide, from some other precursor, then you usually have a linear series of steps. And that is it's a useful concept. It sometimes breaks down. We also have cycles and more complicated things, and we'll look at those as well. But this is certainly a concept that we'll use. And so by pathway, we specifically mean a linear chain. So it's a series of steps going from one metabolite, a substrate or a precursor, to a product. So that's one concept we'll use. But it gets complicated because many enzymes can have more than one role. And if you're looking at a particular protein and you're wondering what it does in your particular setting, it can be tricky. It can have sort of standard annotation as being part of a pathway, but you never know. Some enzymes are part of many different pathways. So it is tricky. But we'll try to get to some concepts to handle this. So I call this course Principles of Metabolism, and maybe it's a lofty name, but we're going to try to figure out some principles that help us cope with this system. So one thing is that there are many enzymes and reactions and pathways, and yes, it's large and complex, but there are only a few classes of enzymes and metabolites, and this I think helps. And we'll look at those and we'll see kind of what patterns there are in these systems. So that's one thing. Another thing is that metabolism must follow physical laws. I'll get back to that a bit later, but you can have some help in understanding the system if you know a bit about how thermodynamics works, how oxidation reduction works, and these kind of things, because it's kind of the same everywhere. All pathways have to follow the same kind of basic ground rules. So that is actually helpful to know. We'll also look a bit at quantitative reasoning, meaning chalking up some basic numbers, doing some back of the envelope calculations of how big is the cell, how much lipids does it contain, if my cells are going to survive on glycolysis, how much glucose should they reasonably consume, things like that. And that helps you to understand measurements, and when you're reading articles and so on, to know if the measurements are reasonable. We'll also do a little bit of modeling. This is towards the systems biology side, you might say. We'll do some flux balance analysis, a very simple version of that, just to get a feeling for how that works. And that can also help, because that can help you sort of in an unbiased way discover things you might not have thought about otherwise. So those are some principles. I'll touch upon a couple of things in this introduction. So one thing that is useful to think about early on is the building blocks. So here I'm putting up a bunch of metabolites that you may or may not have seen. And what I want to sort of look at in these metabolites right now is some functional groups. So there are lots of metabolites, as we said, but there are only a few functional groups. So these are things like alcohol groups, things like carbonyls, which are part of ketone bodies, carboxyl groups, which are part of carboxylic acids, and so on. And if we know something about the properties of these groups, we have a bit of an idea of how metabolites will behave and what they are likely to do. So for example, alcohol groups in sugars are fairly stable, but they are energy rich, and we can extract a lot of energy by oxidizing sugar because of that. Aldehyde groups are usually reactive. If you know that probably a compound like this one is likely to react and maybe give ATP. So that's useful to know. And there are other properties. Amine groups, of course, are things that form peptide bonds in proteins. Thiol groups are something that are very reactive to oxygen, and so they can quickly oxidize, and cysteine, for example, shown here, can easily form cysteine dimers because of this, and so on. And besides their properties, it's good just to know some of these groups, just recognize them as patterns and know their names. I mean, you don't have to know everything, but it's usually good to know some of these names because they come back in metabolite names. So this is tryptophan, for example, and it has as a side chain an indole group. And if you look at the tryptophan pathway and all the metabolites of tryptophan, they're mostly called something with indole. So if you see that name, you can sort of guess that, okay, it comes from there. So that helps you to look at metabolic pathways and learn about them, to know something about the functional groups. Another thing with functional group is that it kind of helps you understand pathways as a sequence of steps. So this is a complicated looking thing. This is purine synthesis, for example. So the synthesis of purine nucleotides. And this is a complicated pathway. There are lots of enzymes. There are all sorts of cofactors and charges and whatnot. But the point here, if you think about this in terms of functional groups, is that you can think about the synthesis of the purine as a sequence of steps where you're kind of adding little little building blocks, little Lego pieces to make these structures. So for example, after synthesizing this phospholipase thing, so the sugar with a bit of phosphate on it, what happens is that you add on an amine group. So that happens here. Everything else sort of stays the same between these two guys, but you add on an amine group. After that, you actually add on a whole piece of an amino acid, the glycine, and then you form this little part attached to the amine group. Again, this has an amine group towards the end. In the next step, you add on something called a formyl, and now you have this, and it's an aldehyde, and so on and so forth. And you can follow these pathways along and see which parts are being added on. And it kind of gives you a sense for how the pathway works. Now, if you would read this pathway in terms of names of the metabolites, it's pretty darn impossible to understand. There are abbreviations here across this whole chart, things like FGAR and FGAM and things, and it's because the names of these things are like half a foot long and they're impossible to read. So I would encourage you not to stare so much at complicated names of things, but look at the structures, and functional groups will help you with that. Another interesting concept that can be useful to understand the larger sort of scope of metabolism is the bow tie model. So this is something, again, that comes from systems biology, but it's a fairly simple concept. And so the idea is that all cells have to be able to convert a lot of different kinds of nutrients into a lot of different kinds of products. So after all, they have to be able to survive in an environment where the diet can contain a lot of different things. And they have a lot of different needs. They need to synthesize a wide variety of different macromolecules and things in order to build their own structures. So that's a tough problem. So the cell has to be able to convert a lot of things on the input side, if you will, to a lot of things on the output side. And if you would have an enzyme for every possible combination of something here and something here, that the number of enzymes that you would need would be huge. So it stands to reason that that's not going to happen. And so what evolution has done instead to organize cellular metabolism is to have something that looks kind of like a bow tie. And well, the shape of this figure is obvious why it's called a bow tie. And what happens is that on the nutrient side, in peripheral metabolism, where you have lots of possible different molecules, you have specialized enzymes that gradually convert these, as this thing sort of narrows, towards a few central molecules. So everything feeds into things like simple sugars or acetyl-CoA or a few amino acids. And then we have central metabolism, and here is where we will spend most of our time during this course. So here you have the most central compounds. You have the TCA cycle, you have amino acids, you have sugars, you have some simple fatty acids maybe. And here you have a small number of compounds that you have to deal with. So you can interconvert this in various ways, but you don't need that many enzymes to do it. And getting from here to here is breakdown of things, catabolism. So you have to break larger things into pieces and make smaller metabolites. And then on the other side, it's the other way around. So here you have biosynthesis of complex products, or anabolism. And here you can perhaps make complex lipids or make glycoproteins or something like that. And once you're back out here, you again have a lot of possible metabolites, many possible products from the cells, and it's peripheral metabolism again. So this is a useful way sometimes to think about when you're looking at a pathway, where is it? Is it a central metabolism pathway, or is it something that deals with a very specialized sort of peripheral thing, like a particular hormone or something that is like only one out of 1,000 possible products? Or is it something very central, like something in the TCA cycle? They will have different properties. So a particular pathway could look like this. For example, you might be able to break down starch, make it into disaccharide, sucrose, a couple of sugar units, make it into something even simpler like glucose, make it into acetyl-CoA, and then maybe you want to start building fatty acids of it, and maybe the fatty acids are used for triglycerides, or so on. So again, we get to the pathway concept. So through this complex network, you can take different paths. So the name pathway, I think, is apt in this case. So the bow tie model you can also make some predictions from this structure. One thing has to do with fluxes, so the flow of material. So when you are on the peripheral side, like on this side where you're looking at the intake of complex nutrients, you typically have many small fluxes. So this is kind of like a capillary bed. You have a lot of different nutrients that are being absorbed, but there's not much of each specific nutrient. So you will have lots of many small material flows on this side. And then when you get towards central metabolism, you get towards this part, you have now entered a few but central metabolites. And you will have a few central reactions, and the flux through these reactions will be quite large. So you will predict that things like the TCA cycle and glycolysis will have a lot of material flowing through them. Whereas something more specialized on this side, perhaps breaking down a particular type of glycoprotein or something, is not going to have a large flux. And this will be the same on the product side as well. So once you're synthesizing a very specialized product of which you don't need so much, there will obviously be a small flux. So this is also good to know when you're thinking about a protein or an enzyme or metabolite that you're studying. Where is it in this system? Are you likely to see large or small enzyme activities? Another thing that you can predict from the bow tie model has to do with how enzymes vary. So if you look at the enzymes in these pathways, when you are out on this side, you typically have a lot of variation between species and cell types. So you might have very specialized enzymes in specialized cells that can break down particular types of nutrients. And you will definitely see that enzymes here will not be conserved to the same extent. You might have something that's specific to human that does not exist in a mouse model, and so on. But when you get to central metabolism, you're getting to more highly conserved, ancient kind of enzymes. These are required by most cell types, so they have stayed the same. If you try to knock one of these down, you're likely going to kill the cell, or you're likely going to kill the organism. You will see a lot of sequence conservation and things like that. And then it's the same when you get to the product side. Again, you will see a lot of specialized things that will have tissue-specific expression patterns, and that will vary between species. So these are some useful predictions that you can make from the bow tie model. One final thing I'd like to touch upon is the difference between metabolism, as I view it, and a lot of the molecular biology that we do. So metabolism, as I have said, it concerns something physical. It's the physical processing of nutrients into biomass and energy and so on. And it's something that really has to be optimized to the physical properties of the environment where the cells live. Signaling and regulation is kind of different. So signaling and regulation, if you think about the growth factor pathway, or if you think about, I don't know, a homeostatic signaling or something like that, hormones, this is all about information. And this is not under the same type of constraints. And I think this makes it easier to reason sometimes about metabolism, because you can kind of say what makes sense and what doesn't. If you know your pathway, you know kind of what amounts of material can be processed by a cell, what numbers are reasonable. When you are kind of in unknown terrain in the signaling world, I think it is more difficult. We don't really know what to expect. And this has to do with the fact that chemical properties are very important for metabolism. So if you see something like this, this is a sugar, you know that this is likely going to be a fuel. It's a good fuel because you can oxidize this to carbon dioxide and get a lot of energy. If you come upon something like this, a purine nucleobase, you will know that this is not a good fuel because it contains a lot of nitrogen. So the chemical properties are very important, and that's why we look at a lot of structures in this course. When you are studying signals or signal metabolites perhaps, or something else, the chemical properties are not really relevant. So we typically don't look so much at chemical structures in other areas of biology. So pretty much signaling metabolites or other events in signaling are just there by convention. So this particular case is adrenaline. And in most species, this means run or fight. It's that kind of hormone. But of course, it has nothing to do with the properties of the molecule or what it looks like. This is just a convention. So the structure is actually irrelevant. This is kind of like Morse code, you could say. It's just a convention that this particular pattern means A, B, C, or D. So again, this means that we can reason about metabolism a bit differently. As we have mentioned, metabolism is under strong physical constraints. This is an example picture of these energy diagrams. We'll look at these later, where you can say something about in which way a reaction is going to move based on properties of metabolites. Whereas in signaling, you really can't say those kind of things. You have to study the system and figure out what it does, but you can't really tell by looking at a metabolite what is going to happen. And this also means that in metabolism, we usually look at quantitative data. Because it has to do with physical properties, it can be very different for a cell. If an amino acid production differs by a factor 10 or something like that, that can mean life or death. So quantitative data will be important, and it's important to look at numbers and think about whether they are reasonable. In signaling or in cell regulation, things like that, it's not as important, I think. We usually look at qualitative data. We look at whether a signal is there or not, whether something has got phosphorylated or not. Maybe we'll look at a bit of relative, twofold up, fivefold down, those kind of things. But we don't look at quantitative data in the same way. So this is, I think, just worth mentioning in this introduction, because I think, for some of you, maybe the course will be a bit different in that we'll look a lot at chemical properties and a lot at quantitative data. Okay, so that's it for this first lecture.